Now here's another problem on the net charge of the peptide. So in this case, I have a 5 amino acid peptide. So I have cysteine, valine, lysine, arginine, and glutamic acid. So these are the 5 amino acids in a peptide, and I want to calculate the net charge on this pentapeptide or 5 amino acid peptide at pH of 11. So I told you in last video when we did a calculation on the net charge for a tripeptide for 3 amino acid peptide, I told you that we need to consider two things that are really important. So one is that we only have one C terminus to consider, the C terminus of the very end amino acid. So in this case, glutamic acid, and the N terminus on the other end on the starting amino acid of the peptide chain, in this case, cysteine. So we only need to consider the charges or pK values on these two terminal, N terminus and C terminus. The rest of them, as we know, are peptide bonds, right? So we don't consider the charges for all of them. So that's a really important first point. So once you get that out of the way, you consider the pK values at a certain pH that you're given for these two terminal uh, amino groups and carboxy groups. Then you can go on to calculate the R groups pKa values, the pK values for R groups. So in this case, we'll consider only lysine, arginine, and glutamic acid. Why? Because only these three amino acids are charged. Cysteine and valine are neutral, right? As we know that. But I told you, cysteine and tyrosine are the only two amino acids that are not charged at physiological pH, around physiological pH, that we need to consider because their R groups can be ionizable, right? So I'll show you what that means. So apart from basic amino acids, lysine, histidine, and arginine, and acidic amino acids, aspartic acid and glutamic acid, cysteine and tyrosine are the only two left that we consider in when we are calculating net charge calculations, okay, for at a certain pH. So let's go ahead and calculate the net charge on this pentapeptide. And here we are given a basic environment, right? We are given a pH that's well over the physiological pH of 7.4, a pH of 11. So we know that we are dealing with basic environment. So let's first tackle according to the first point, let's first tackle the C terminus and N terminus. Okay. So we know that for C terminus, our pKa value is 2. And for the N terminus, it's 10, right? So we know that pH of 11 is higher than both of their pKa values of 2 and 10. We know that both will be in what form? In deprotonated form, right? Why? Because according to the Henderson-Hasselbach equation, right, as I showed you in the last video, we know that if pH is greater than pKa, we know that the amino acid will exist in the deprotonated form, right? Right? That makes sense. So we know that these two N terminus and C terminus will be in their deprotonated form, right? So we already know that the C terminus is already in its deprotonated form, but we need to go ahead and deprotonate this because in a basic environment, the OH minus ions will accept. So base, you know, they accept protons, right? Their Bronsted-Lowry bases, they accept protons, right? So this will be deprotonated. So now we have neutral charge on the N terminus. Now we only have H2N sequence here, right? So we don't have any charge on the N terminus anymore. So overall, we have a negative charge for these two terminal groups to consider, the N terminus and C terminus. So C terminus is negative, N terminus is neutral, so we have a net negative charge. So we are done with the first step. Now let's go on to the second step. Now we need to consider the R group pKa values. So of interest, we can ignore valine over here because valine is, we know that it's a neutral amino acid, right? So we don't need to consider its R group, but we'll consider the rest of the four. So let's start with the cysteine one. Okay, so for cysteine, I wrote the pK values over here in this table. Cysteine's pKa value for its R group is 8.33. So same trick, we know that pH 11 is greater than 8.33. So cysteine will also exist in the deprotonated form, right? So again, according to that rule, since pH is greater than pKa, we will have the deprotonated form predominant over the protonated form, right? So cysteine will be deprotonated, right? So in that case, it will lose its proton from its the SH side chain bond, right? We know that cysteine has a sulfur containing side chain because it's involved in sulfur disulfide bridges, right? Cysteine is involved in disulfide bridges. So cysteine has its SH group, and it will lose its proton and it will become negatively charged, the sulfur present on its R group, right? So cysteine will now, I'll write the R group as for cysteine as a negative charge, okay? So it will become negative. Valine we don't need to consider. Lysine now will be, let's see what lysine will be. So lysine has a pKa of 10.79. Now this is also smaller than the pH 11, right? So pH is greater than the pKa for lysine, so we'll again have a deprotonated form over here. But be careful, for lysine, its deprotonated form is not negative because basic amino acids like lysine, arginine, and histidine, they already exist in the positively charged form in their neutral configuration around physiological pH. So once they lose their proton, they will become neutral instead of negatively charged. So from positive, they become neutral. So this for lysine, I don't need to put any charge on that. Okay, same argument for arginine, but careful because arginine has a pKa value of 12.48, which is now greater than the pH. So now we need to do the reverse thing. So now for arginine, it's the opposite case. Because its pK is greater than pH, now it will exist in the protonated form, right? So it will stay in its positive configuration, right? If we had a pH of 13, then obviously it will be greater than the pK value for arginine's R group. Then we'll have arginine also as a negatively charged, as a neutral charge, right? Now for glutamic acid, that's the last one we need to consider. Now it is an acidic amino acid, which means its R group in its neutral configuration around physiological pH exists in a negative form, right? So for glutamic acid, we have a pKa value of 4.25, right? 4.25. Now, its pK value is very less than the pH, right? As we can see over here. So pH is greater than the pKa value for glutamic acid. So it will exist in the deprotonated form. Okay, same as it was for lysine and cysteine. Okay, so since it will exist in the deprotonated form, it will be negative, right? Because if it was protonated, then it will lose its negative charge. It will accept a proton, right? It will get a proton. In that case, it will be protonated, which will be only found in the acidic environment. But in basic environment, it will still exist as a negative charge because there are no protons around there. Okay, so this is our whole peptide sequence for this 5 amino acid peptide sequence with charges. So we have done the problem. We have considered the C and N terminus, 2C and N terminus, and we found that N terminus is neutral, but the C terminus carboxy group is negatively charged, and we also considered all the R groups except valine because that's neutral. Okay, so now we can find the net charge. We can see that there's one negative, another negative, and another negative, but a positive charge. So we have 3 negatives and a positive. So we have a -2 or net -2 charge. So that's your answer at pH of 11. Okay, so that's how you tackle these kind of problems. All you need to do is that you always start with the number 1 rule, which is you first consider the C and N terminus and their pKa values. Once you have found those 2 values and found that if they are positive, neutral, or negative, once you're done with that, then you can go on to consider R group pKa values. So if you have in your peptide sequence that you're given these 7 amino acids, only then you will consider R groups. Otherwise, if they are all, if they're all, let's say, like valine, methionine, isoleucine, or alanine, those other, except these seven um, amino acids, then you don't need to consider R groups. But if you have these seven amino acids, then you go ahead and consider their R group pKa values, right? And then once you have all that, you can find the net negative, net negative or positive, whatever it is, net charge on the entire peptide sequence. Okay. So I hope that was helpful, and I think I made that really easy.